Imagine a tiny structure inside every cell that can recycle waste, fight invading bacteria, and even trigger the cell's self destruction. Can you guess who it is? Yes, you are right. It is none other than the lysosomes. So, meet the lysosomes, the cell's cleaning crew, the cell's garbage disposal unit, the cell's emergency and demolition squad. It can digest everything, so it does all in one function. It is small but powerful, and it is mainly responsible for keeping the cell healthy, clean, and safe. So let us learn more about this wonderful cell organelle. Are you all ready? So let's learn. So here we have the lysosomes, which are very small in size, and as I said, big in function. They perform a very big function, a very vital role. They help in digesting the waste, they help in recycling the old parts of the cell, they help in defending the cell, that is killing the bacteria and all, and they maintain cleanliness inside the cell. So they are the cell's garbage disposal units. So, lysosomes are membrane bound organelles. They are made up of a single membrane, and they are filled with, as you know, because when they are doing all these functions, there is something inside the lysosomes which is doing all these work. So what is that magical substance present inside the cell? It is nothing but the digestive enzymes. So these digestive enzymes, they are responsible for performing all these functions and protecting the cell. The lysosomes, the cell's cleaning and recycling systems. Now, if you see, the lysosomes are small membrane bound vesicles present in the cytoplasm, and they are, as I already told, they are made up of a single membrane. Now, the lysosomes are also known as pericanalicular bodies. So, they are also known as pericanalicular bodies. So, you should remember this term. Sometimes the other words are also asked in exams. Pericanalicular bodies. So this is the other dense bodies. So this is the other name of lysosomes. And in plants, children, lysosomes are also known as spirosomes. I'm sorry. Right. So lysosomes in plants, they are also known as spirosomes. Now, these lysosomes, they contain, as I told you, they contain something magical inside them. So what is that magical? That is nothing but hydrolytic enzymes. Remember children, the meaning of the word lytic. So lytic, when we use the word lytic, lytic means breaking. So the hydrolytic enzymes, their main function is to digest the waste materials, worn out cell organelles, and other foreign substances. So that is the main function of these hydrolytic enzymes, and for the first time they were discovered by a scientist called as Christian de Duve in 1955. Now, when you talk about the occurrence, they are abundantly present in RBC, liver, pancreas, bone, cartilage, kidney, thyroid, epidermis, where there is lot of secretion taking place, and they are absent in mature mammalian RBCs. As you know, in mature mammalian RBCs, the cell organelles are absent, including the nucleus. They are abundant in phagocytic cells. Phagocytic cells, the cells which play a major role in eating away the foreign bodies, and the meristematic cells in plants which help in cell division. Right. Now, if you see the structure, as I told you, it consists of a single membrane bound organelle, and they contain rich fluid, and that is the enzyme called as acid hydrolases, which is mainly responsible for breaking down these substances. And the membrane, now when there is this acid hydrolases or the hydrolytic enzymes present inside, right, we can have a doubt like how is the cell being protected. So why, how all the other cell organelles in the cells are being protected from these lysosomes when it has such a dangerous substance present inside? So the answer is the membrane. So the membrane which is present on these which covers up the lysosome, which we generally call it as a tonoplast. So this membrane is responsible for protecting the other cell organelles or protecting the whole cell. So the membrane protects the enzymes from digesting the cell itself. Now if you see, the lysosomes are very small, so they vary from 0.2 mm to 0.8 micrometers, and the lysosomes are found in the kidney cells and WBCs. These hydrolytic enzymes may rupture under some conditions like injury, disease, chemical exposure. So in such cases, the membrane of the lysosome may rupture, and the rupture releases enzymes which can cause a breakdown of the its own cell. That is what we call it as autolysis, and that is the reason why the lysosome is also known as a suicidal bag of the cell, because it is responsible, once its membrane ruptures, it has the ability to eat away all the other cell organelles of its own cell, right? So that is why it is known as the suicidal bag. Now let us talk about the stability. When we said that about the stability of the lysosomes in healthy conditions, in normal conditions, the lysosomes, they are maintained by the hormones called cortisones, corticosterones, and cholesterol. That is, as long as these hormones are there, the lysosomes are kept in, they are maintained in their healthy condition. But the moment there is some default in these hormones, or there is some diseased condition, the lysosomal membrane is ruptured, and the moment the lysosomal membrane is ruptured, right, all the hydrolytic enzymes come out and they may eat away all the other cell organelles, and this process we call it as autophagy. Remember children, phagy means eating, auto means self, eating one's own cell, right? And this can be done artificially by injecting the hormone glucagon. So when the hormone glucagon is injected, then also the lysosomal membrane will rupture. Now, the lysosomal membrane also ruptures in the presence of vitamin A, E, X-rays, bile salts, and hormones like progesterone. So, they also have the ability to rupture the lysosomes, and also lack of oxygen also causes a breakdown. Now, there are four types of lysosomes. So, this will be a better one. So, the primary lysosomes, the secondary lysosomes, the autophagosome, and the residual body. Now, let me explain this in a very simple form. Now, primary lysosomes, imagine a bar, a washing bar, which is covered with this the plastic cover or the paper cover what it comes. Now inside you have the rinse soap or the washing soap with its cover. So this is the original bar, or we call it as primary lysosome, which contains the lytic enzymes, right? So it is formed from the Golgi lamellae, right, and Golgi membrane, and you have the primary lysosomes with the hydrolytic enzymes, right? Now next, the moment you remove the cover and your bar is ready, the washing bar is ready, now the washing bar comes in contact with any foreign body, any foreign body. So when the primary lysosomes come in contact with any foreign body ready to digest it, that we call it as secondary lysosome. So the secondary lysosome is a functional part. Now here the secondary lysosome, so the foreign substance, it can be in the form of phagocytosis or pinocytosis, which phago is cell eating and cell drinking. It can be in the form of a liquid, or it can be in the form of a solid. Now the plasma membrane forms a vesicle or brings all those things to the lysosomes. Now it forms the secondary lysosome. it is ready to digest. That means our soap is ready to clean. Its cover is out and it is starting to perform its function. Now, what is this autophagosome? Children, auto means self. Auto means self, phago, eating. Now in the cell, if there is any cell organelle like the mitochondria, endoplasmic reticulum, which is which is worn out, all those cell organelles are directed towards the lysosomes, and the lysosomes will engulf its own cell organelles of its own cell. That we call it as autophagosomes. So digestion of intracellular substance, which we call that, the process we call it as autophagy. Clear? So this is a third type of lysosomes. And last type, residual body. Now lysosomes will destroy everything which is present inside them, right? Now when it destroys, there are certain things which it cannot eat up, it cannot be destroyed, so those are converted into residual body, that is the undigested debris, right? So this undigested debris which is present in the lysosome, we call it as a residual body, and this is eliminated by exocytosis, or what we call it as aphagy, right? Okay, so these are the four types of lysosomes. You should remember the primary lysosomes, the secondary lysosomes, the autophagosomes, and the residual body. So you can see this is a rin bar with the cover, and the rin bar with cover removed, and the it is coming in contact, the washing process is going on. And here the autophagosome means the lysosome will digest its own or worn out cell organelles, and the undigested debris is which is not digested, that is sent out, eliminated by exocytosis, right? So now here you can see the primary lysosome here with all the hydrolytic enzyme. So when the primary lysosome is coming in contact with a foreign body here, undigested matter, it becomes a secondary lysosome. And you can see here the primary lysosome here, when all the worn out cell organelles are sent towards it, it becomes autophagosome, and the undigested matter, again you can see it is stored in the residual body, right? Now if you see the chemical composition, the hydrolytic enzymes or the acid hydrolysis, they contain some enzymes called proteases, which is a cathepsin and collagenase. It contains acid carbohydrases, right? The alpha glucosidase, alpha mannose, alpha beta galactosidase, and beta glucuronidase. So these are the acid carbohydrases enzymes. And the acid lipases are acid phosphatases and acid lipases, and other enzymes. So all these acid sulfatases, acid DNAs, acid RNAs, these are all the acid hydrolases, the enzymes present which make up the hydrolytic enzymes present in the lysosomes. And they also contain ferritin granules. Ferritin granules are nothing but the iron containing proteins, which also form a very significant part of the hydrolases, right? So now next, when we were talking about the digestion pathway, there was one very important part which we have learned is autophagy. Children, autophagy is it is one of the most the discovery, or the most very interesting discoveries so far. It is on autophagy. So let me tell you about this autophagy. It is nothing but the cell digesting its own cell, the lysosomes digesting its own cell. Now here lysosomes use heterophagy. Heterophagy means, here you can see, which is coming from outside. So now this is a lysosome. One more thing I want you all to remember is lysosomes work under the pH of 4.5 to 5, that is acidic condition. So they work under acidic medium, right? So now here if you see, the lysosomes, they use heterophagy to digest external materials, and autophagy, where they engulf and recycle their own damaged organelles. Like you see, anything coming from outside, that is pinocytosis or phagocytosis from outside, whatever bacteria or other substances are there, they form the early endosome, turning into late endosome, and those are sent into the lysosomes through endocytosis. And the lysosomes will they help in the wearing out of all these substances. And then anything which is inside the cell, so inside the cell, that those all cell organelles and all, these cell organelles are worn out cell organelles are destroyed, and this we call it as autophagy, and this becomes the autophagosome, right? So just remember that uh, phagocytosis or heterophagy is nothing but which is coming from outside the cell. Autophagy is where the cell, where the lysosome is recycling or engulfing its own damaged cell organelles, right? Then next, autophagy, it helps in removing the damaged cell organelles. Autophagy helps in removing the damaged cell organelles. It also helps in recycling the cells during starvation. So recycling the nutrients during starvation. And it also helps in fighting infections. You might have heard these days that fasting is very important. So why? Because during fasting, all these enzymes, they become active and they fight infection, they help in recycling of nutrients, and they prevent the accumulation of toxic substances. They prevent the accumulation of toxic substances, right? And they maintain the healthy cells and tissues. That is why starving or fasting is considered to be very good for your health. And now they maintain healthy cells and tissues. And problems in autophagy, when a cell fails to perform the function of autophagy, then it may result in cancer, it may result in Alzheimer's disease, Parkinson's disease, diabetes, or aging related disorders. And the recent exciting discovery of about autophagy, one of the recent exciting discovery is that they are not only lysosomes are not only garbage bins, but they are the control center for cellular metabolism and nutrient sensing. So they help in sensing of nutrients. So here you can see the lysosomes, they maintain the cholesterol homeostasis, right? So they, through cell signaling, they maintain even the lipid metabolism in the cell, and they also help in the repair of plasma membrane. So these are one of the very important points related to lysosomes. So autophagy is nothing but, autophagy is the cell's self renewal system. So it is the cell's self renewal system. Autophagy is a cell's self renewal system, right? So now let us move on to the next one, the functions of lysosomes. We have mostly covered. Heterophagy means extracellular digestion. You can see whatever comes from outside through cell eating or cell drinking, phago or pinocytosis. So like bacteria, food particles, they are all digested, right? Autophagy, intracellular digestion, all the worn out cell organelles, you can see, right? Then it also helps in intracellular digestion and recycling. All the old cellular components, they are all recycled, and the useful substances are again released. And autolysis, that is a self digestion. So when the lysosomal membrane ruptures, the enzymes are released and the whole cell is digested, that we call it as autolysis. And I told you that is the reason why the lysosomes are also called as suicidal bags of the cell. They are also known as the suicidal bags. And because they are destroying everything, they mainly help in defense mechanism, right? So you can see how they are destroying the harmful microorganisms and protecting the cell. Now the diseases caused due to abnormal activity. So when lysosomes stop performing their functions, so it results in accumulation of undigested material, and that results in a disease called as Tay-Sachs disease, Gaucher's disease, and Niemann-Pick's disease, right? So these are all the diseases when the undigested material is accumulated in your cell. And the poor lysosomal activity also results in infections, where the cell's ability to destroy bacteria and all is very poor, incomplete digestion, and that it leads to frequent infections when pathogens are still present in your body. And because the lysosomes, they help in autophagy, destroying the worn out cell organelles. Imagine when the lysosomes function is not performed, the cell organelles are not destroyed, and that may result in abnormal lysosomal functions. It may help in cancer cell survival and invade other tissues. These cancer cells may increase in number and they may invade other tissues, right? Tissue breakdown and invasion, because the cell's main function of eating away the worn out cell organelles and autophagy is affected due to lysosomal malfunctioning. So it may result in cancer. Then inflammation, the rupture of lysosomes, the rupture of these enzymes, it will damage the surrounding tissues. So because these enzymes are very acidic enzymes, very harmful enzymes, when these are sent out, this results to inflammation and cell injury, and they also result in genetic disorders, right? So the lysosomal enzymes may cause enzyme deficiency, which results in metabolic disorders. They may help in gene mutation, that means sudden change in your genetic organization. So these are all the diseases caused due to abnormal activity of lysosomes. Now let us move on to what we have learned. Let us revise. The enzymes present in lysosomes work best at neutral, acidic, alkaline, or variable pH. As we have already learned, it is 4.5 to 5 pH, that is acidic. So the answer is acidic pH. Which of the following is not a function of lysosomes? Lysosomes will digest the foreign particles, they break down the damaged cell organelles, they help in intracellular digestion. Not the function is ATP synthesis, which is done by mitochondria, right? So the answer is C, which is ATP synthesis. Now, if lysosomes burst inside a cell, they may cause. So when lysosomes, this is a lysosome, when they are bursting inside, so these autolytic or the hydrolytic enzymes, they will eat away its own cell. And that process we have learned, it is called as autolysis. Auto means self, lysis means break, right? Now select the correct statement regarding lysosomes. They are formed from Golgi apparatus, this is correct. They contain digestive enzymes, this is also correct. They are double membrane bound, they are not double membrane, they are single membrane, right? They help in autophagy, this is correct. So 3 are correct, 1, 2 and 4. So the answer is B. Clear? So hope you have understood the most important points of lysosomes to be remembered. See you in another episode of BioBytes. Keep learning. Thank you.